I'd like to thank Professor Magdi Yaqub for inviting me for this and to address a meeting which is dedicated to taking molecular science into man and then onto the community. And I think this is exactly describes how Professor Magdi Yaqub himself has progressed in his career. And I'm only partially got there because I started out as a biochemist. And I've always wanted to get my work into man. And I think I'll show you through this talk that it is possible to get from molecules to man, although we haven't made it into the community yet. And part of the reason for this is this collaboration which I've managed to establish between our group in Bristol and a group of epidemiologists led by Gerard Pasterkamp in Utrecht. And it's been with the support of the NHS National Institute for Health Research. I'm grateful to Dr. Falk for having introduced the subject so well for us. We know that most myocardial infarctions are caused by plaque ruptures, which occur at the narrow shoulder region of the plaque where the cap is thin. And if you look at a lot of these plaques, as he said to you, they tend to be bigger plaques. They tend to have a thin fibrous cap, less than 65 microns. They have a large lipid core. They're inflamed. They have more macrophages and T cells. And he made this important point that the core of the plaque is depleted in collagen. And in fact, the thin cap itself is depleted both in collagen and in the cells that produce collagen, vascular smooth muscle cells. And he also made the point that the inflammation that you get in plaques is slightly different from other types of inflammation because it involves foam cell macrophages rather than conventional macrophages. He also introduced you to the matrix metalloproteinases, and these are the molecules I'm going to talk about. Increasingly, people are taking a system-wide approach to studying molecules. We've seen some talks about this already. It makes things more complicated for audiences and for us scientists. So I hope you'll bear with me because I'm going to show you a kind of system-wide approach to studying the role of MMPs in atherosclerosis. And I think what it really shows to you when you do this is that you shouldn't really think of metalloproteinases as all being the same. There are some very different properties for some types of metalloproteinases rather than others. And therefore, the pathogenesis for individual matrix metalloproteinases is different, and by implication, we should try to target the ones which are of most pathological significance and not the group of MMPs as a whole. I want to dedicate the rest of my talk to two other pathologists, Michael Davis, who's on the left of this picture, two British pathologists, I might add, who back in 1992 asked me a question that I couldn't answer. And he said, can you explain to me why when I stain for anything other than CD68, only some foam cell macrophages are positive? And the other British pathologist I want to talk about is Simon Gordon, who has helped in a major way to provide the answer to this question. Basically, what Simon Gordon has dedicated the last years of his career to was looking at the diversity of macrophages in inflammatory conditions. And basically what he showed was that when monocytes are attracted into sites of inflammation, they can differentiate into macrophages. In fact, there are two major cytokines that mediate this differentiation. One's called MCSF and the other one's GM-CSF. And actually, there's a whole story about the diversity between those two populations, but I haven't got a chance to go into that today. But basically, what Simon pointed out to us was that when these mature macrophages encounter a pro-inflammatory environment, which is dominated by molecular patterns like LPS, which come from bacteria, or from coactivators like CD40 ligand, or interferon gamma, which is coming from T cells, or inflammatory cytokines like interleukin-1, which is coming from the inflammasome, or other inflammatory cytokines like tumor necrosis factor, which can be produced from endothelial cells or damaged smooth muscle cells. Under all these set of conditions, the macrophages tend to differentiate into a form we call M1, or classically activated. And in an infectious site, these macrophages become more invasive. They're more able to undergo phagocytosis to remove the bacteria, and they're also better at killing bacteria. On the other hand, if macrophages arrive in an anti-inflammatory environment dominated by interleukin-4 or interleukin-13, they can differentiate into a much more helpful wound healing type of macrophage called M2, or alternatively activated. And these macrophages are good at getting out of lesions, they're good at antigen presentation, they're good at promoting fibrosis, calcification, and angiogenesis, which is part of the wound healing response. Now, there are two major points I want to address in my talk, and one is that how relevant is this to atherosclerotic plaques? Well, we don't have conventional macrophages, but we have foam cells. And secondly, is the adverse effect of these macrophages, or foam cell macrophages, attributable to only one or other of these phenotypes? So let's deal first with the idea that what happens when you take up oxidized LDL and become a foam cell. And if you argue simply from logic and what's happening in the plaque, you might think that foam cell formation is actually harmful. In fact, you can read a whole mass of literature suggesting that oxidized LDL makes macrophages more inflammatory. The truth is actually that it makes them less inflammatory. And several studies recently have shown this, but this is the best one. And this was done by an enormous consortium of researchers led by Chris Glass and published this year in Cell. And basically what they showed was that when macrophages become foam cells, one of the things that happens is they start to produce a lot of this sterol cholesterol precursor called desmosterol. And in fact, the reason for this is that the enzyme that converts desmosterol into cholesterol is downregulated when you take up oxidized LDL, and this leads to the accumulation of desmosterol. And desmosterol is an activator of LXR, which is a nuclear hormone receptor, and this brings about a whole range of changes in the macrophage behavior. And one of the crucial things that happens is that it downregulates responses to pro-inflammatory stimuli like toll-like receptors, and this is shown on this genomic study here. Oh, good, that actually works, wonderful. So you can see all this mass of genes that are normally upregulated by toll-like receptors are actually downregulated by foam cell formation. And importantly, all of these experiments were done in mice, but he did actually show that the same thing happened in human foam cells. Okay, so foam cell formation itself is not responsible for the pro-inflammatory effect or the damaging effects of macrophages in plaques. So there must be something else responsible for it. So we can go back here. So we know that, in fact, foam cell formation seems to be anti-inflammatory. So the question is, is this scheme actually even valid for foam cells? Can you actually polarize foam cell macrophages into these classically activated and alternatively activated states? Well, the simple way to prove this is simply to make some foam cells in vitro and then treat them with these various ligands to see if they will actually polarize just like macrophages. And that's what I've done in this experiment, which is very colorful, a bit complicated, so I'll go through it slowly. So basically what we do is we take something, this is cyclooxygenase 2, and it's a very good marker for classical activation. And classical activation so occurs in normal macrophages, as shown by the upregulation by 1,000 times of cyclooxygenase 2. And it also occurs if you've previously made the cells into foam cells. So those foam cells are able to polarize into classically activated macrophages. Why do they do this? Well, they do it because they activate a transcription factor called NF-kappa B. And this is a readout for NF-kappa B, and you can see in the normal macrophages and the foamy macrophages, we get upregulation of NF-kappa B. Okay, what about the metalloproteinases? Well, some of the metalloproteinases are also upregulated under these conditions. So this is one particular one, it doesn't really matter, it's called MMP14, and that gets upregulated with classical activation, at least by a factor of 10, but not by alternative activation with interleukin-4. So it's following the pattern of cyclooxygenase 2. What about alternative activation, or M2? Well, we do that by just adding interleukin-4 to the cells. And if we take a marker for alternative activation, CD206, we can see that this occurs in the normal macrophages, and it also occurs to the same degree in the foamy macrophages. Now, this is a very interesting phenomenon because CD206 is actually downregulated by classical activation, as well as being upregulated by alternative activation. So when we look through the metalloproteinase system and also the tissue inhibitors of metalloproteinases, we did actually find some molecules which obeyed this alternative activation pathway. So in particular, MMP12 here, it obeyed the alternative activation paradigm, and also TIMP3. So as I'm going to go on to show you, most of the metalloproteinases actually follow this pathway. But TIMP3 is actually being downregulated. So we've got an upregulation of many of the metalloproteinases, but the inhibitors are downregulating. And this suggests that indeed in foam cell macrophages, they become more proteolytic as they go towards the M1 pattern of activation. Now, we don't need to go through this slide in any great detail, but the previous set of data I showed you was messenger RNA data. So the question is, do you get the same phenomena if you actually look at the proteins? It's obviously important to show that, and the answer is yes, we do. So for example, when we look at MMP14 or COX-2 proteins, they get upregulated by M1 conditions. On the other hand, if we look at TIMP3, CD206 or MMP12 proteins, they do indeed get upregulated by alternative activation. Okay, so the next question we went on to ask is, well, what particular inflammatory mediators are important for generating this pattern of activation? So what we've done in this experiment here is to compare the effect of LPS, which is an activator of toll-like receptors, with TNF and interleukin-1, which are two well-known cytokines, and which come from the tissue, but also with the interferon gamma, which is a cytokine which is particularly coming from T cells, which may be also in the lesions. And what we find is that several metalloproteinases, as I told you, will get upregulation under M1 conditions. So in addition to MMP14, we've got this MMP1, MMP10, and one which I'm not going to show you on this slide because it's got such low activity in the absence of stimulation that you can't actually calculate a fold stimulation for it, and that's MMP3. And MMP1, MMP3, and MMP10 seem to act together in a very simplistic and a very simple M1 pattern. MMP12 is a bit of a Jekyll and Hyde, because you can see when you look at it in detail, it does get upregulated by LPS, but it gets, as well as being upregulated by IL-4, as I told you in the previous experiment. And interestingly, it's quite sensitive to interferon gamma as well, which a minority of these proteins are actually sensitive to interferon gamma. But perhaps the most interesting finding on this slide is actually that of all those activators that I've been looking at, the only one which will inhibit TIM3 is interferon gamma. So this suggests that in order to get this fully evolved pattern of more MMP14 and less TIMP3, you can only do this if you have T cells present in lesions that are producing interferon gamma. And that is quite an important conclusion as it happens. I'm not going to go into this in any great detail, but there are some very well characterized signaling pathways which are responsible for these various events. As I said, the production of the transcription factor NF-kappa B is a particularly important event. Activation of the PI3 kinase pathway and activation of these extracellular receptor kinase pathways, which activate this transcription factor called AP1, are also known to be intimately involved in the pro-inflammatory signaling. And in fact, when we used inhibitors of any one of these pathways, which is basically what is shown on this slide, that all of these inhibitors of each of these pathways would downregulate the production of MMPs of this type, showing that in reality, all of these MMPs are in fact inflammatory mediators. If you look at the table of inflammatory mediators, you see all kinds of things on them, but you rarely actually see MMPs. listed there. But for a number of years, we've been trying to get over this idea that MMPs are, in fact, inflammatory mediators. The only pathway that seems to have a minor importance here is this P38 pathway. Okay, interferon gamma signals through a different pathway completely, through something called Janus kinase 2, and it produces a transcription factor called STAT1. So the question is, were the interferon gamma effects being mediated through this pathway? And the answer is, again, I'm not going to go through this in detail, but both the ability of interferon gamma to activate these several MMPs and to inhibit TIMP3 are dependent upon the activity of JAK2. So what I really want to get, the simple conclusion I want to get over to you is that MMPs of this type are as good inflammatory mediators as anything else. When we surveyed the whole metalloproteinase system, I've been focusing on this little group here, we could see that the pattern emerges that many of the MMPs are in fact activated under M1 inflammatory conditions. And the only ones that ever come up under M2 conditions are MMP12, TIMP3, the inhibitor, and MMP11, and several other metalloproteinases are not really affected at all. The other thing that's on this slide, and you can see it in this paper if you want to go back to it, it actually gives the relative abundance of these proteins too, so you can see which ones are most important and least important, at least at a bulk level. Okay, so all of that was in vitro. What about taking this stuff in vivo, into man? Well, when we take these observations into a human biobank of specimens, we can actually show that there's a co-localization of MMP1 and MMP10, in fact MMP14 too, with COX2, which is the marker of classical activation, and with nuclear localized NF-kappa B, the transcription factor. So this confirms that in human lesions, and indeed in foam cell macrophages in plaques, the conclusion I've been telling you is still valid, that classical activation is what's causing the production of these molecules. Okay, and that's just quantification of the data I showed you on the previous slide. Okay, so we got a very simple conclusion, which is that normally foam cell macrophages have rather low levels of metalloproteinases and high levels of TIMPs. But when you get classical activation by a number of mechanisms, you get upregulation of a whole string of metalloproteinases. Okay, but is there a relationship between stability and instability or vulnerability of plaques? Well, first of all, I want to show you some data, in vitro data, which we got from a rabbit model, where we could actually make foam cells in vivo and extract them from lesions. And actually what we showed was that these foam cells we got from the rabbit had exactly the same pattern of upregulation of these several metalloproteinases and downregulation of TIMP3. And then we could take these foam cells and look at their behavior in vitro. And what we could show is that there are actually two populations in here. There are ones which lack TIMP3, and there are ones which have some TIMP3 left in them. And when we look at the ones that lack TIMP3 in this section, you can see that they appear to be more invasive in the lesions. And when we took them out and put them in tissue culture and conducted a simple experiment to see if they would actually invade through a layer of synthetic matrix called matrigel, we found that only the MMP14 positive and TIMP3 negative ones would actually migrate through the matrigel layer. So basically, the ones that had this pattern were more invasive. And in fact, they have more ability to break down matrix, which is what we showed in this assay. And something we hadn't expected, these cells are also more likely to undergo apoptosis. When you think about it, these are exactly the properties that you'd expect for the type of macrophage which would destroy an atherosclerotic plaque. They're more invasive, they get down to the base of the lesions better, they're more destructive, they're getting rid of the collagen from the core and the cap, and they're also more likely to die and therefore contribute to the acellular debris that you get in the plaque core. Okay, well that's one kind of data. But then we wanted to go on from that rabbit data into actual human data, and this is where we set up this collaboration with Gerard Pasterkamp, and he has more than 2,000 biobank samples. They're mainly carotid specimens, and I'm only going to show you the carotid data now. Basically, this is a very powerful methodology because he has all the patient characteristics of these people who've had carotid endarterectomy, but also he has follow-up data on all the patients as well. So we can do cross-sectional studies where we relate the characteristics of plaques to the presence of MMPs, but also we can relate the presence of MMPs in these plaques to what happens to the patient subsequently. Okay, so I'll show you some of our cross-sectional data, and Professor Falk made a very important point, which is that the actual volume of the plaque that's occupied by inflammatory cells can be really small. So what we actually do in all of these experiments, we actually compute the ratio between the presence of these MMPs and the presence of CD68. So even if there are only a few macrophages in a specimen, we can get a proportion. And basically what this data shows is that the proportion of macrophages which express MMP12 or MMP14 is higher in vulnerable plaques, thin cap fiber atheromas, than it is in stable plaques, these pathological intermill thickenings. And this is also true for the marker of classical activation, COX-2, whereas conversely, the presence of TIMP3 shows the opposite relation, just as the marker of alternative activation does, CD206. Okay, and in fact, if we dissect this more in detail, we find that all the characteristics you associate with stable plaques, such as the amount of collagen, the amount of smooth muscle cells that are in the plaque, they negatively correlate with MMP14 and positively correlate with TIMP3. And if you take all the things that are characteristic of unstable plaques, like how big the lipid core is, how many macrophages are in them, these positively correlate with MMP14 and negatively with TIMP3. And moreover, if you actually look for what's causing MMP14 to be upregulated, again, it's closely co-localized with this marker for the activation of NF-kappa B, whereas if you look at TIMP3, it's closely co-localized with CD206, which is a marker for the alternatively activated macrophages. Okay, so in a cross-sectional study, clearly activation of macrophages, classical activation of macrophages correlates with vulnerability to rupture. And it looks like alternative activation might correlate with stability. Okay, what about the outcome for the patient? Well, we did actually look at MMP14 levels, TIMP3 levels, and MMP12 levels across a group of 236 patients. You need a lot of patients to do this study. I'm not going to tell you about MMP14 and TIMP3 because it didn't prove they didn't prove to be a relationship. And I think the reason for that is technical, because MMP14 and TIMP3 are not only produced by macrophages, they're also produced by smooth muscle cells. And so it's difficult to be sure that you've actually localized it correctly. So MMP12 was the one that came out most clearly, because it actually is only produced by the macrophages in the plaque. So what we did was we took these plaques from 236 patients. We had 3-year follow-up data. We looked for any vascular event or intervention. We stained for MMP12 and CD68, and we calculated the ratio, and then we divided the patients into quartiles, depending on the percentage of MMP12 positive macrophages they had. Okay, and this is what the quartiles look like. So there's about 60% positivity in the top quartile, and no positivity at all in the bottom quartile. And then when we did a Kaplan-Meier analysis, we could see that there was actually a statistically significant benefit in terms of survival, such that the ones which had no MMP12 had a much less likelihood of having a clinical event than the ones that had a high level of MMP12. So the simple conclusion from this is if you've got too much MMP12 in your plaque, you're more liable to die afterwards. Now, this is not just an artifact of there being more macrophages in the more MMP12-rich plaques, because if you actually look at the CD68 staining, you can see that this has no relation to subsequent survival. So this is another very important point from my talk, is that it's not the total number of macrophages in the plaque that matter. It's the number of macrophages that are expressing particular phenotypes. And we want to go on and do this with the other MMPs that we showed related with classical activation, particularly MMP 1, 10, and 3. But we haven't done those studies yet. That's work in progress. And when we looked at the specific endpoints, well, obviously, these were mainly strokes in this patients which had carotid disease. Okay, so too much MMP can kill you. That's the message from this. Okay, well, that would be a really bad message to end on if it weren't for the fact that we showed last year that a very selective MMP12 inhibitor can actually arrest atherosclerosis. Okay, this is a mouse study, but it does suggest to you that if you can design a reagent that specifically interacts with MMP12, that you can actually do something about this adverse effect. Okay, so we come to the conclusion that indeed, when you classically activate macrophages, you get a whole bunch of MMPs being produced, and that this does indeed lead to a more vulnerable plaque phenotype. Okay, now, MMP12. Now, what I showed you earlier on is that MMP12 is a bit of a Janus-faced enzyme because it can be also upregulated by alternative activation, not just by classical activation. So it's a bit ambiguous. So we wanted to trace down the regulation of MMP12 a bit more. And alternative activation can be distinguished from classical activation because it works through a different transcription factor pathway. Instead of being regulated by NF-kappa B, it's regulated by STAT6. And when you upregulate MMP12 using IL-4, we could show using this technology here, which was actually adenovirus-mediated delivery of an inhibitor, that you can actually block the production of MMP12 under alternative activation conditions, either at the RNA level or the protein level, using this blocker of phospho-STAT6. Okay, so when you're doing it under alternative activation conditions, you can indeed show that it works through this STAT6 pathway. But when we went back into the plaques and looked for the activity of STAT6 in the plaques by using an antibody which labels the activated form of the STAT6 transcription factor, phospho-STAT6, we couldn't actually find any phospho-STAT6 in the plaque. The only place we could find it was in the adventitia. So this is nuclear phospho-STAT6 in the adventitia. But we couldn't actually find any in the plaque. So although we've got very good evidence for classical activation of macrophages in plaques, we don't have any evidence for alternative activation at this stage. Okay, so most of the macrophages in plaques seem to be classically activated, and they're the ones which are causing the problems anyway. So let's forget about alternative activation. But is classical activation the only thing which will upregulate these proteins? Well, that experiment we got on that previous slide encouraged us to start looking for other things that might upregulate MMPs. And we started to do some experiments with hypoxia, because we know that there is a lot of hypoxia. Thanks to Matt Darman's work, we know there's a lot of hypoxic macrophages. And actually, when we started to survey the metalloproteinase system under hypoxia, we did find that hypoxia will also upregulate MMP12 in a STAT6 independent manner. And I haven't shown it on here, but it also upregulates MMP1 and MMP10 as well. And basically, what we've been able to show subsequently is that there is a tendency for these metalloproteinases to co-localize together with the transcription factor which characterizes hypoxic cells, which is called HIF1 alpha. So I'm not suggesting that hypoxia by itself upregulates these MMPs, but hypoxia does seem to cooperate with classical activators to give you higher levels of these MMPs in the plaque. So the reality is probably that it's a synergy between classical activation and hypoxia of macrophages within the plaque, which is particularly inimical. So basically, this is my final conclusion here, which is that if you just have unactivated foam cell macrophages, they don't tend to have a lot of TIMP3 and not much metalloproteinases. And this is a stable situation. There don't seem to be many alternative activated macrophages in plaques that we can find. But if you classically activate them, you get a lot of metalloproteinases, you reduce the TIMP3, and you get a vulnerable phenotype. And if the macrophage, it's the final statement. And if you have hypoxic cells, this also further regulates the metalloproteinases and leads indeed to poor prognosis. And then that is my final slide, which is just to say thank you to my colleagues and to the British Heart Foundation, which supports me.
Thank you very much indeed, Professor Newby. This paper is now open for discussion. Can I ask you one question, and that is, what are the therapeutic implications of all what you have told us? I mean, you mentioned 3 things which we could manipulate, M1 and M2, for example. The second is MMPs, and the third is JAK-STAT pathways. Can we really manipulate that? And if you are willing to predict which way we are going to go finding a solution to a difficult problem?
Definitely. Now, just sticking to MMP12 for a moment, I showed you that there is a selective inhibitor there. So obviously we're very keen to develop that, and we have got a collaboration going with Astra looking at an MMP12 inhibitor. Okay. There's another interesting thing is that we showed that MMP12 actually has prognostic value in the plaque. So we might actually be able to use this to get, and it is possible to use substrates of MMPs to generate a signal in the plaque that you can detect externally. So this could actually turn out to be a marker. So that's just looking at MMP12. Now, when it comes to classical activation, one of the things we've been very, very keen to do is to find out what the specific role of interferon gamma is in this, because this has been hypothesized for many, many years based on mainly on animal data. But actually, when you go back and look at the human data, there's incredibly little human data to support it. But if interferon gamma is really important, and we do think that it is important in the human plaque, then we can use immunomodulation. And so basically, we can immunize people against the antigens like oxidized LDL, which are a major source of the inflammation which is in the plaque. And because this would be specific to a particular antigen like oxidized LDL, there's a much better chance of hitting this adverse kind of inflammation compared with just blocking inflammation in general. So far, when people have tried just to block inflammation in general, it does reduce the coronary events, but it actually causes problems elsewhere in the body. You get more tumors, you get more infections, and so on. So we need to be more sophisticated than just looking at blanket anti-inflammatory treatments. Now, whether or not specific agonists like interleukin-1, which are being looked at with an antibody trial whose name has just escaped me, whether specific pro-inflammatory agents or anti-inflammatory agents like antibodies to interleukin-1 will be effective, that's being tested out at the moment in a clinical trial.
Thank you very much. One more question? Yes, sir. Dr. Peter.
So Paul Ruptured has launched a cardiac inflammation reduction trial, CIRT, a big NIH proof of concept trial. This will use low-dose methotrexate on top of statins in high-risk patients for a next cardiac event. Is there any data so far on methotrexate or some of these other, especially the newer biologics you developed for immunotherapy, rheumatoid arthritis, Enbrel, other types of medicines? You know where I'm going on this, because this is the next, and these agents work very fast. Is there going to be a role for those potentially in ACS patients, or I would think very high risk post-MI patients?
You would introduce agents just for mechanobiology?
Hope went into the idea of using PPAR gamma agonists, which are anti-inflammatory agents that hit these pathways. And as you know, the history of these trials was that you actually didn't reduce overall mortality with these drugs. So my prejudice is to think, obviously, like most clinical trials, these are, at the end of the day, are empirical trials of a concept that's been developed by basic science, and it may turn out that for reasons which you don't fully understand, that you do get an overall benefit from a general, one specific type of overall anti-inflammatory agent. But my prejudice from the science would be that you have to be more specific than that you have to be more selective than that.